Assalamualaikum and hi. We are from Group 1. My name is Nurafa and my groupmate is Nasima and Nur Hidayah. So for today's presentation, our topic is the use of microalgae in bioremediation of the textile wastewater. So this is the detail for our main journal article. In textile industry, 8,000 of chemicals are used for the whole process. The waste from this industry can give a negative impact to natural water if it is not properly discharged and managed. In 2018, it was stated that 5 billion kilograms of chemical are used to produce 49 billion of clothing. The usage of chemical in textile industry has brought many applications, especially to natural water bodies. With an improper management of waste, the waste can end up in the nearest river and contaminate the soil. Hence, the release of textile wastewater to water bodies become the main concern of the researchers in this paper. From the paper chosen, the purpose of the study is to investigate the effectiveness of the microalgae in decolorizing the dye. This is a new technology that has been used to handle the contaminants from textile effluents. This technique, also known as bioremediation, as it uses the biological agents such as algae, bacteria, and fungi to eliminate the toxic substance and heavy metal in environment. The specific question in this paper is the ability of microalgae in treating the textile wastewater effluent. To identify the ability of the microalgae, the experiment has been done by the writer of this paper. There are three parameters that are used to identify its ability. First is the growth of a microalgae. Second is the ability of the microalgae to remove the red color. And the third one is the ability of the microalgae to remove the heavy metal. The use of bioremediation in managing the textile wastewater effluent can help to lower the negative impact by the chemical use in textile industry. The chemical use in textile industry can affect humans by causing allergies and birth defects. This is due to the carcinogenic and mutagenic substance in the effluents. Hence, bioremediation is the best solution as it is considered as a safer, eco-friendly and cost-effective technology to decontaminating the pollutant.
Hi, I'm Nasima and I'm going to explain about the 5 methods that were used for this study. For the first method, 5 microalgae strains were obtained. It is selected based on their ability to develop, decolorize and remove heavy metals from the tested textile wastewater effluent. The tested microalgae strains were grown and maintained in a growth chamber under white continuous illumination at a temperature of 25 plus minus 2 degrees Celsius on BG11 medium. Next is by applying 200 ml of textile wastewater effluent that were kept in a 500 ml Erlenmeyer Meyer flask and individually inoculated with 10 ml of each microalgae strain, then tested for the various properties such as water reaction, pH, electric conductivity EC, dissolved oxygen DO, and electrical conductivity EC together with BOD and COD. After incubation for a period of 28 days, each filter was subject to the weighing procedure and exposed to a solution of DO using UV visible spectroscan 21 for spectral decolorization. pH and EC were also measured to assess the microalgae weight at constant oven dryness while the filter papers were in the oven. The decolorization percentage is as shown in the slides, whereby the initial absorbent is subtracted with observed absorbent times 100 initial absorbance, and the unit is in percent. For physicochemical analysis, the pH, EC, DO, COD and BOD levels in the microalgal treated TTWE filtrate were measured. It is measured as using the following formula. D1 stands for the dissolving oxygen after sampling, while D2 is the DO level after 5 days incubation at 20 degrees Celsius. P is the decimal volumetric fraction of the sample used. heavy metal analysis, whereby the atomic absorption models that were created in England was used to calculate the heavy metals amount at TWWE. The following slide is the results for the study that represent the highest reduction percentage growth is for Anabina floss aqui, which is 19.37, and the lowest growth is for Polrelia vulgaris. Also, it is observed from the physicochemical study that the DO level is low and the BOD is high following the COD level, which is very high, and the salinity level and also the pH level. And it can be concluded that a lot of heavy metals were found and the area is highly polluted.
What was particularly striking about this study and its finding? First was the study concluded that Nostoc ellipsosporum has the greatest results with highest dissolved oxygen DO, highest percentage removal of chemical oxygen demand COD, biological oxygen demand BOD, and removal of color from the red textile wastewater. The 100% removal of color means the microalgae decompose 100% azodized for its nutrient requirement. The highest biomass means more bioabsorption, with each microalgae has storage pools for heavy metals, which then resulted in the highest percentage reduction of heavy metal. Second finding is that the 5 microalgae are mesotrophic, a combination of autotrophic and heterotrophic culture. The 5 microalgae are autotrophic because they are capable of photosynthesis. The photosynthesis causes increased oxygen concentration, which will then be used in decomposition of organic materials. This can decrease COD and BOD as shown in the main journal. The journal then mentioned that they were azodized at the discharge point and thus they have azogroups which contain nitrogen. The microalgae will then degrade dyes for nitrogen source to grow. This means that the 5 microalgae are also heterotrophic. As the 5 microalgae are mesotrophic, the growth rate, biomass production, and lipid productivity is 1.74, 14 and 5.6 times higher than the microalgae with autotrophic growth only. From the main journal, the microalgae biomass produced can be a great renewable feedstock for biofuel production because of its high lipid yield. Plus, it is the most economical alternative of feedstock in producing third-generation biofuels. Third-generation biofuels of microalgae are devoid of major drawbacks that the first-generation biofuels had, such as arable land, high use of fertilizer, pesticide, and water. Also, third-generation biofuel is more economical than the second-generation biofuels that need to include pre-treatment, which is costly on a large scale. The question that this study did not answer is whether pH is affecting the ability to remove dye. The study has shown the correlation between amount of biomass and initial dye concentration towards the discolorization by microalgae, but did not investigate whether pH affects removal of color too. Iski and CB in 2019 said that the pH did indeed affect the dye decolorization efficiency, with the optimal pH between 6 to 10. Nostoc ellipsosporum, Nostoc linkia, Anabena variabilis, and Chlorella vulgaris recorded 179, 88, and 96% of color percentage removal, respectively. However, Anabena floss aqua recorded a much lower color percentage removal, that is 50.81%. Thus, the textile wastewater pH of 9.5 is assumed to be unsuitable for Anabena floss aqua, and thus the experiment should include pH as manipulated variable to see whether Anabena floss aqua have higher color removal percentage during lower pH. In the study, magnesium, calcium, and sulfate were listed along with other initial physico-chemical characteristics of the untreated textile wastewater, but there is no mention for their treatment nor the results because it cannot be reduced using microalgae. But it can be treated in tertiary treatment that remove the final contaminants in wastewater after the secondary treatment using microalgae is done. The treatment opted is ion exchange for magnesium and calcium, and reverse osmosis for sweetened sulfate. The main journal also wrote that the azodyes that were used in textile wastewater were degraded by microalgae into simpler aromatic amines, but the product is hazardous to the aquatic environment and mutagenic for humans. As the azobond is easily broken under anaerobic conditions, it can cause aromatic amines residual which can be degraded under aerobic conditions. The solution is then using a simultaneous anaerobic-aerobic system, as the aerobic process can immediately degrade the aromatic amines.
That's all from us. Thank you.